and Pierce. This is Dr. Jones checking in. This is ANP1 lecture test 4, video 3, and we're on Noteset page 4, which for me looks like this. So the sarcolemma. I told you that sarcoplasm would come back to haunt us, right? No, not sarco. Well, no, I said it wrong, didn't I? Okay, erase. All right, plasmalemma. I liked the blue though. We still blue? Excellent. The plasmalemma is a cell membrane. So this is the cell membrane of a muscle cell. And muscle cell membranes are special because they are excitable. You got it. You got it. Sarco for flesh, lemma for husk. So flesh husk. That's disturbing. So it's like the sausage casing. It's the flesh husk that surrounds your muscle cell. Yeah, that's how that works. So it's a phospholipid bilayer like every other cell membrane, and it has what makes it different, what makes it excitable, is that it has voltage-gated ion channels. To see. So the voltage-gated ion channels are what allows the sarcolemma to be excitable. Okay. The sarcoplasm then is the cytoplasm of flesh. So sarcoplasm is just cytoplasm, but it's special because it's a muscle cell, and muscle cells are excitable. It's all about the jazz hands. It's all in the jazz hands. You got it. Okay, so the transmembrane potential, as we reviewed in the first video for lecture test 4, refers to the fact that it's relatively negative charge inside the cell and a relatively positive charge outside the cell. You got it. The change in the transmembrane potential is called an action potential. Got it. Okay, action potential. The change in the transmembrane potential is called an action potential, and is the first step that leads to a muscle contraction. And then in bold with 2 sets of asterisks. Changing the transmembrane potential is an electrical activity roughly analogous to flipping a light switch. I got 2 little lights over here. It's a daylight and the big light's on, so that didn't help much. Which affects voltage-gated ion channels. True. So the voltage-gated ion channel works when you change the voltage. When you change the voltage, like that. Let's see if I can reach the closer one. There we go. Okay. Voltage-gated. Normally I stand on the side of the room and flip all the light switch for a few minutes until everyone's giggling. Then we get back to class. The muscle action potential is carried inside the cell by the, we already said all of this, didn't we? Sarcolemma, sarcoplasm, transmembrane potential, muscle action potential. voltage gain. I wrote that down. I didn't have to write that down. Okay. So what carries that muscle action potential inside the cell is the transverse tubules, which are sometimes called T-tubules. So here they are. You can see it's actually cell membrane that has grown inside the cell and makes little tubes inside the cell. And they have, as their lining, as their edge, the membrane is the same stuff as the sarcolemma, which means the T-tubules are, jazz hands, excitable. So they carry the muscle action potential into the cell. Why? That allows, the muscle action potential is what makes the muscle fiber contract. So it allows the entire muscle fiber to contract at the same time. Simultaneously means at the same time. And it's the same stuff as the sarcolemma. It's the brain fart bilayer phospholemma. Oh my god. It's got the head and the tail. Okay, it's too late in the day for my brain to work. We'll move on. Bilayer. Help me out here. Put it in the comments. I know I said it in the last video. I had it like 10 minutes ago. It's just poofed out of my head. Things do that sometimes. Usually during the test. Okay. So that's all about transverse tubules. Now let's talk about the sarcoplasmic reticulum, the SR. So the sarcoplasmic reticulum is specialized smooth endoplasmic reticulum, and it surrounds each myofibril, which we haven't learned about yet because they changed the order of the sections. And it stores boatloads, and I do mean boatloads and boatloads and boatloads of calcium. Like there's 40,000 times more calcium inside the sarcoplasmic reticulum than in the sarcoplasm. Ooh, and let's make that distinction here. This is sarcoplasm, the cytoplasm of the cell membrane. And this blue stuff is the sarcoplasmic reticulum, the SR, spell it out for lab, which is specialized smooth endoplasmic reticulum that's specialized to store calcium. Okay. And there are enlarged portions of the SR touching the T-tubules. And these are called terminal cisternae. Terminal means end, and a cistern is a storage chamber. So these are storage chambers of even more calcium next to the T-tubules. And we'll talk about why. I don't think right now, but we will talk about why. Okay. And the two terminal cisternae and the one T-tubule between them form a triad. So here's the triad. It's all the way across. You've got the T-tubule and the two terminal cisternae. So like a tricycle triad. Okay. And the SR functions to store calcium ions, which is Ca++. Or Ca2+, describe the relationship between the calcium ions and the muscle contractions. Okay, here's where I tell you. Okay. The excitement, the muscle action potential of the sarcolemma, the book often shows it as a red lightning bolt. Okay, so the muscle action potential travels down the length of the, that's a zigzag lightning bolt, of the sarcolemma, the cell membrane of the muscle cell. And then it also travels down the T tubules. And what is each T tubule touching? 2 terminal cisternae. So the trick here is that the terminal cisternae have voltage-gated calcium ion channels. So when the excitement reaches the T tubules, the excitement flips the switch on the voltage-gated calcium ion channels. Let's flip it on. There we go. And opens the calcium ion channels. Now, we know that the sarcoplasmic reticulum and the terminal cisternae have 40,000 times more calcium in those little tubes than there are in the sarcoplasm surrounding the little tubes. How much calcium do you think is going to come out? Like a water hose, okay? It's like going to flood the inside of the cell with calcium. And that calcium is going to bind to stuff that we haven't learned about yet. So I'm not going to give you the terms and confuse you. But it's going to bind to stuff, and it's the calcium binding to stuff that's going to allow the muscle to contract. How's that for vague? We'll clear that up in a video or 3. All right. So the muscle action potential travels down the sarcolemma and down the T tubules and opens the voltage-gated calcium ion channels in the terminal cisternae. And then, like a firefighter water hose, lots of water, lots of calcium. You release the dogs, the calcium. And then there's calcium everywhere inside the cell. And it's the calcium binding to specific proteins that allows the muscle to contract. And that's how you move. There's more to it, but we'll add details. Okay. So I do want to say, I don't know if I said it, the sarcoplasmic reticulum, and it looks like a sweater sleeve to me, and then the terminal cisternae are then the cuffs on each end of the sleeve. The terminal cisternae are part of the sarcoplasmic reticulum, just like the little hemmed up cuff is part of your sleeve. It's just kind of a specialized part of the sleeve that has its own term. It's still part of the sleeve. The terminal cisternae is still part of the sarcoplasmic reticulum. It's specialized, and so it has a specific term. It's part of the triad. It stores even more calcium, and it has those voltage-gated calcium ion channels. So, oh, other thing, sarcoplasmic reticulum surrounds each and every sarcomere. And now let's talk about myofibrils to get the rest of the information together. The muscle fibers contain hundreds of thousands of cylindrical, rod-shaped myofibrils. And it's the rod thing. And they are the length of the muscle. Myofibrils consist of bundles of crystalline protein filaments called myofilaments. Myo means muscle. So myofibrils are made of myofilaments. And we've mentioned the myofilaments before. They include thin filaments, which are made mostly of actin, and thin filaments, which are made mostly of myosin. And we talked about actin and myosin are myofilaments. and that they're in groups called sarcomeres. And we'll talk about that. And the interaction between the actin and the myosin, the interaction between the thin filaments and the thick filaments, the interaction between myofilaments are responsible for muscle contraction once calcium binds to a specific protein that we'll learn. We haven't learned that yet. Okay. I think we have a quiz slide. Oh, when the myofibril shortens, the entire cell shortens, and that pulls on the tendon. So this right here, this is your biceps brachii, and it ends here at the biceps tendon that attaches to the forearm bones, one of the forearm bones. And so when the muscle shortens, because that's how muscle contracts, it gets shorter. When the muscle shortens, it pulls on this tendon, biceps tendon. It pulls on the tendon, which is attached to the bone, and that's what flexes your elbow. And those terms, some people use it to say flex the joint, flex the elbow, and some people say to flex the body part that moves. So some people will say flex the forearm, but it's the same thing. Flexing the elbow, flexing the forearm, same difference. Okay. Quiz. I think this one's first. What are the little rods in the muscle fiber cells called? Oh, it's not going to show us the answers? What happened? Okay, so what's this? Myofibril. What are the edges here? Sarcolemma. What's the cell stuff inside? Sarcoplasm. And what are these oval seedy bits? Those are nuclei. Remember, it's multinucleate with multiple peripheral nuclei. The inside of the cell is all these myofibrils and a boatload of mitochondria because you need energy to make muscle cells go. Okay, more quiz. What are these? Mitochondria, because you need ATP for muscles, lots and lots. What's the cell membrane of a muscle cell called? Sarcolemma. The rod thing is a myofibril. Now, one of the nice things about having lecture and lab books from the same publisher is that they use the same color coding. So for our publisher, the red lines in the muscle cells are going to be thin filaments, and the blue lines in the muscle cells, muscle fibers, are going to be thick filaments. Okay, and then what are these three together? 3 is for triad. The sweater sleeve is the sarcoplasmic reticulum. And what about these yellow tubes? T-tubules. T-tubes is not for credit in lab, not even a little bit. It's got to be tubule. Huge distinction, makes a difference. And what are these cut bits? More myofibrils. And then the cell stuff, sarcoplasm, cell membrane, sarcolemma. And then the part of the SR that's touching a T-tubule, terminal cisternae. There you go. Okay. This has been A&P 1 lecture test 4. We're on page 4, so it must have been video 3. I haven't stuff. There it is. Okay. All right. Thank you as always for tuning in. The education must go on. Dr. J out.